Avi Mayan.
Hello, I'm representing the Links Data Coordination Center, which is also a BD2K center. I have one scientific highlight and then a list of contributions. So the connectivity map at the Broad Institute stimulated 10 core of diverse human cancer cell lines with over 20,000 small molecules applied different concentrations, and then they measure gene expression using the L1000 technology at different time points. So in total they generated over 1 million gene expression profiles, and that data potentially can be a transformative contribution that can transform pharmacology and drug discovery. We, the BD2K Links DCIC, developed a statistical method that we believe extract more knowledge from that data. So this method identify better differentially expressed genes, and the reason it works better is because it's a multivariate methods, and currently methods such as Lima or DCIC are essentially univariate. And we process all the links L1000 data using this method and then benchmarked it against the method that is used at the Broad to create signatures. And here I'm showing that we were able to identify a drug structure similarity from gene expression similarity. And this also allowed us to benchmark different methods that compute drug structural similarity. So now we have a method in hand that we can use to better extract knowledge from this L1000 large data set. So we developed a web-based tool, a search engine, that can be used to compute the angle between gene expression signature that a user would input and query it for drugs that can either mimic or reverse expression of a specific condition. So in a conversation with Chris Bassler, who is a professor at Mount Sinai, a microbiologist, what he's interested in is identifying interferon alpha mimickers as potential antivirals. And this is how we apply this method now to identify several novel antivirals that are compounds that were profiled by Links. And the way we did it, we collected signatures from GEO, where interferon alpha was applied to different types of human and mouse cells. We also processed interferon alpha signatures collected by a joint project between the HMS Link Center and the Broad Transcriptomics Center. And surprisingly, many of those experiments, whether they were done using different platforms or using the L1000, they all reported the same drugs as potential mimickers. Those drugs were uncharacterized drugs, and they were shipped from the Broad and from NCATS to Chris, who validated them. So when Chris called me and told me that those drugs actually worked, I was not too surprised, because the overwhelming evidence that we collected to show that those mimickers work across cell lines and assays. So what type of contribution our center is making and will continue to make? One effort that we have, and we have a poster on it, is, thank you, we collecting data from public resources and organizing this data to create gene set libraries and attribute tables for machine learning. As I've shown you, we are big on creating benchmarks to improve computational pipelines and also extract more knowledge from data and address issues like reproducibility. We also develop metadata and ways that would make it easy to submit data sets and link those data sets to ontologies. We have collected what I believe the largest set of annotated gene lists, and also collected from the world, which we're going to keep private, over 500,000 of submitted gene lists to our tools. We also delivered 2 MOOCs courses on Coursera and had over 33,000 registered students. And we also running some crowdsourcing projects, and those are microtasks and megatasks. And one of the projects is to extract and annotate gene expression signatures from GEO. And we also released our first mobile app on the Google and the App Store, and this allows you to find information about genes and protein from all of those resources that we are processing. We also have collaborations with 2 of the BD2K centers. One is with the Cedar Center about collecting and coordinating collections of metadata, as well as with the NoN Center trying to integrate data sets from links with other data sets for machine learning and other processes.